Okay, there we go. Let me just do my introduction. Okay, so hi, my name is Siddharth Nair. As part of my grief support advocacy initiative, I'm compiling a collection of conversations to capture expertise, experiences, and the science between the two. As part of this series, joining us today is Dr. Nathaniel Johnson, assistant professor at the Department of Nutrition and Dietetics at the University of North Dakota and vice president of the North Dakota Nutrition Council. Thank you for joining us today. Um,
Well, thanks for having me.
do you want to talk a little bit about like what you do?
Oh, yeah.
So,
I mean, I'm an assistant professor of nutrition and dietetics. Uh, I study the link between dietary intake, mental health, and disability is where I do most of my work. Um, I started off my dissertation, if for folks who are curious about that, was about dietary protein intake, muscle health, and disability. And specifically, we were looking at dietary protein intake distribution. So that'd be somebody's protein intake at breakfast, lunch, and dinner. And what we found, and it was what we had expected to find, is that having a more even distribution of dietary protein was related to better muscle health. Um, in a sample of women aged 18 to about 80. So it was a really big sample group of women. And that's what I
mhm.
did my dissertation on. And it was, rewarding and kind of set the stage. When I moved up from my old institution, which was also in North Dakota, to now UND, I started to change things a little bit because I didn't have the same access to the same kind of research equipment that I had in my previous institution. And so I started to focus on mental health, dietary intake, and disability. And in tandem with that, a topic area that comes up a lot would be food security. So I do a lot of work related to food security and how that can impact stress. And on that note is how we get into the realm of epigenetic related research. So nutritional epigenetics actually stems from work that looked at famine or exposure to famine, and that would be by definition food insecurity, and then its effects on DNA methylation patterns. Now, DNA methylation patterns on their own are kind of a rascal to understand and wrap your head around because depending on where that methylation occurs in a gene, it can either upregulate or downregulate the expression of the target gene. So just because there's a methylation on that area doesn't mean that we know right away or just cursory glance at it whether that gene target's going to be upregulated or downregulated. Now, when we get into the really, the details, you will be able to know that, but it depends on the type of DNA methylation analysis that's done. And so epigenetics and DNA methylation is a naturally occurring and ongoing process. So aging itself, right, causes epigenetic changes, and the epigenetic genome or the epigenome can be associated with changes in aging as well. So, in other words, you could use the epigenome to estimate somebody's molecular age, if you will, which is an interesting notion. You can also combine that with the telomere shortening to also estimate somebody's age from a molecular standpoint using their genetics. Um, and as far as questions that I'm interested in, is there's a very clear link. These group of researchers from Brazil linked food insecurity status to depressive symptoms. So they actually found out that it mediated through this gene methylation patterns, in particular the NR3-1C gene, NR3, NR3C1, that's right, gene, and that then was associated with different depressive symptoms. So my question, and this also relates to grief, is that stressor, is experiencing food insecurity as a younger person, like a child or an adolescent, excuse me, then going to result in not just a lifetime of differences in DNA methylation pattern, but also DNA methylation patterns that might be passed on to, well, future generations. There's a group of colleagues of mine who do work out of UW Madison, and in their animal model, they found that sheep fed an amino acid just during adolescence and one generation resulted in epigenetic changes that were not only persistent throughout their life, but were passed on at least two generations down. So that means that a stressor like food insecurity or a stressor like grief is going to have potentially not just a lifelong effect, but an effect that goes beyond generations. And so, in my own research, this is a powerful motivation for addressing food insecurity. People don't like to, generally speaking, give other people food, which I find to be unfortunate. And that doesn't just have implications for the health of the person you're choosing to feed or not feed right now, but that person's kids and their kids. And so we got to start thinking about food and food access, and also in the case of grief and other adverse childhood experiences, as having lasting effects that go beyond just that person and then cut across generations. So it's a
Hmm.
big deal. And as far as future research projects, we're working on a animal study to look at food insecurity, which is a hard thing to emulate in the animal world because people and how we approach food is a lot different than any animal. Um, so there's limitations there, but we like to look at food insecurity, its effects on the epigenome, and the plasticity, which I think is something you want to talk about today, Sid, which is how much can these be changed,
Hmm.
and the result that once a negative stimulus is removed, or there's some treatment for it, can we actually change some of these DNA methylation patterns and then improve health?
Hmm. So I think one thing to cover, like before we start diving really deep, is like, could you like briefly explain like what DNA methylation is and like the general role in the human body?
Okay, so DNA methylation is the addition of a methyl group to a DNA sequence. Methyl group is a carbon with three hydrogens attached to it. The methyl
Hmm.
group, depending on its location within that DNA sequence, whether if it's on a promoter region where we have these um, in this case, they'd be transcription factors binding that can inhibit that. But if it's put later down in the stream, it can enhance the actual transcription area of a particular gene. So where the DNA methylation occurs is actually important again for its upregulation and downregulation of that gene. Now, when I mean upregulation or downregulation, that can occur across multiple steps. So keep in mind that the vast majority of our phenotypes or outward traits are actually determined by the proteins that are produced in our body. So,
Hmm.
like, right, melanin's a great example of that. Sid, you got more melanin than me, which is
Hmm.
why you're darker than me, right?
Hmm.
And
Yeah.
if you have albinism, if you're albino, then you have no melanin in that
Hmm.
case. Well, that's just one example of phenotype and then how it can be relate to genotype. But what we have here is that your DNA is going to be transcribed into messenger RNAs that are then translated into proteins. So change in DNA expression can also mean a change in the transcriptome. That's the production of messenger RNAs, or it can be actually a change in the proteome, the actual number of proteins that are produced. Now,
Hmm.
really to have the biggest effect on outward phenotype would be to have a change in the concentration of proteins and not just a change in the concentration of messenger RNAs.
Hmm.
But upregulation would be increased messenger RNAs, and downregulation would be decreased messenger RNAs, which would then correspond to increased expression of that protein or decreased expression of that particular protein in that gene sequence.
Okay.
Now, there are gene sequences that don't code for proteins, but those are more well, number one, there's quite a lot of junk DNA that doesn't encode for anything. But as far as other coding sequences, there are some DNA sequences that encode for like Pee-wee interfering RNA, microRNA, oh, sorry, Pee-wee interacting RNA, excuse me, and small interfering RNA
Hmm.
are also both coded in our genome and also regulate expression as well. So, for instance, certain microRNAs might bind to messenger RNAs, preventing them from being translated into proteins, thus augmenting gene expression.
Hmm. So I know you touched on earlier the gene NR3C1, I think, but in your opinion, do you think there are any like specific genes or biological pathways that are regulated by DNA methylation that you believe could be particularly relevant to like the biological and psychological process associated with like grieving?
That's a tough one, Sid.
Hmm.
Um, Uh, I mean, as far as specific genes, I'm not going to be able to give you a specific gene. Now, I would guess that, and I need to look it up to make sure I have the right numbered name for it, but I'm going to guess that NR3C1 gene, which is implicated in depression, would be one of the main targets of epigenetic related regulation. Um, One of the more challenging aspects currently of, I would say, the studying the epigenome in general is that we can look at DNA methylation patterns, but right now that's kind of where the buck stops. So we're looking at like Usually what's presented in research is you have a gene and then we have the percentage that's either hyper or hypomethylated. So hypermethylation would be increased methylation relative to your control or your target group. Hypomethylation would be decreased. And so, although on an individual level you can kind of sort some of this out, the issue here said is on a physiological or on a specimen level, one change is likely to result in a whole host of epigenetic changes. So it's
Hmm.
not just one thing that happens or one gene that's probably affected, it's a whole series of them. So when
Hmm.
we look at like the data from the Dutch hunger winter, it's the entire genome that's affected. And so it gets harder to pick out which exact uh, locuses of those genes we want to focus on as far as like mental health. And keep in mind the complexity, right, of mental health and like the grieving process. Um, and other aspects of our physiology, it's easier to say, like, well, there's only a couple genes that encode for this or whatever. But probably a lot of different proteins and aspects of physiology go into good mental health. And so I don't think that there's going to be one specific gene or like set of genes that you would normally look at. Now, um, if you do some genomic work, like essentially the basis of some of this work would be to take people out in the population and take people who have a certain condition or don't have that condition and then compare their methylation patterns. So, for example, you could say something like, and I'd have to find more data to send you in the right direction, but let's just say, one group of people, they have depression. They might have increased methylation of this particular gene relative to your healthy group, if you will, without depression. And so that gives us an indication from a research standpoint that that gene's probably involved in that relationship. And so establishing the exact functionality, Sid, of these genes and their role in mental health and stuff is very complicated. And I just don't think that there's specific target genes that I mean, you could look at anything. Let's put it that way. But I don't think that there's one gene that's the all maker as far as mental health. You'd have to have a pretty complex and holistic look at those different genes that are being affected. And you'd have to relate them, like I said, to differences in the methylation patterns that we see in populations where we know they have these conditions.
Do you think there could be like different DNA methylation patterns for people who experience grief at a young age versus people who experience grief at like an older age?
Definitely. So the epigenome research suggests that the sooner the adverse experience, the larger the effect on somebody's physiology and thus epigenetic markers. So the
Hmm.
Dutch hunger winter research is from people who were in gestation. They were in their moms during the Dutch hunger winter. So they experienced it, but I mean they experienced it as a fetus, which does count from a food security standpoint, but they don't remember anything about it, right? And so, yeah.
Hmm. So like how about like changes in DNA methylation patterns following like a significant loss contribute to biological changes? I know you like touched on like the idea of like the protein expression. But I guess I was wondering specifically, like, do you think there could be like removal of DNA methylations, like the reversal of those
Well, that's
ideas?
that's exactly right, Sid. Is that you got to remember DNA methylation is used as a regulatory process,
Hmm.
and so the general structure of the previous set of research, and this is what I mean by we don't know a lot about the overall effects. We have relationships, but the actual mechanism is more complicated or less elucidated would be a better word to put that. Um, but let's put it this way: when we looked at the DNA patterns from um, the Dutch hunger winter, what they did is they compared siblings who experienced the famine to those who didn't. And then they looked across a variety of different genes looking for both hypermethylation and hypomethylation patterns. So hyper would be extra methylation of that gene relative to the sibling. Hypo would be decreased methylation of that gene relative to the sibling. And so it does happen in either process, and either one can actually be a negative or a positive. So all hypermethylation isn't good or bad, and all hypomethylation isn't good or bad.
Hmm. I see what you're saying. So, do you think like I guess one thing that I looked at with one of my science fair projects was like maybe trying to reverse DNA methylations. And I know we've talked about this earlier, and you kind of mentioned it with like the plasticity. So could you like talk a little bit more about the plasticity, I guess, of DNA methylations?
Yes, and on that note, Sid, I got bad news for you in the sense that my colleagues over at UW Madison, who did the transgenerational study,
Mm-hmm.
their basic conclusion was that these are not very transient, meaning they're not very plastic. The changes seem to be relatively permanent. Now, that's an indication from one study, which is why we need to follow it up with another more robust research using animals, right? But unfortunately, the current data seems to suggest that once the damage is done, that there might not be that much we can do to reverse it. And so it's hard. That being said, like when we talk about grief and a major loss, that's a little bit different than these experimental paradigms we've been using for investigating uh, DNA methylation patterns. So, specifically, what I'm saying here is that when we do the DNA methylation pattern and when those researchers at UW Madison did their study, they actually altered diet during all of adolescence.
Mm-hmm.
Now, when you experience a loss, now I know people hold that with you, but how long does that actually affect somebody's physiology? And in the paper I just sent to you, looking at adverse childhood experiences and increased age of somebody's epigenome, so essentially DNA methylation patterns that look more old, would be the way to
Mm-hmm.
put that, look associated with older people. They found that people needed to have 4 adverse childhood experiences,
Okay.
which would suggest to me that you need a fairly stable or a fairly long period of time where people are experiencing that adversity or that consequence to have the DNA methylation pattern. Now, we don't know that, just to clarify, we don't know that, but
Mm-hmm.
that's what that suggestion would give me. And so when you talk about grief, there's a couple of things. One would be limiting the number of adverse childhood experiences, and the second one would be decreasing their duration, right? Because
Mm-hmm.
if they're shorter in duration, there's less odds that you see these DNA methylation patterns that right now don't look like there's actually that much we can do to change them.
Mm-hmm. Mm-hmm. I see. So do you think like you touched on a little bit, I like do you think there's a way of like looking at DNA methylation patterns and being able to like identify like, oh, this person has gone through grief whereas this person hasn't? Is there a way to like identify that through DNA methylation patterns?
I would say that hypothetically, yes,
Mm-hmm.
but practically speaking, I don't think we have anything like that developed. Um, so what I'm trying to say, Sid, is that practically speaking, or hypothetically, yes, you should be able to look at somebody's DNA and say, with this methylation pattern, we give this probability that they're had an adverse childhood experience or something like that. With we need some supporting data to do all that sort of calculation. But
Mm-hmm.
in order to actually have the statistical power to make such predictions, you'd also need a very large data set. And that's where I was saying, like, practically speaking, you probably can't do it because the data is not out there. I don't think we have a data set where we have enough people who've gone through grief, um, where we can have kind of this is the DNA methylation pattern of these people, right? And then use
Mm-hmm.
it as a testing marker for somebody who comes in brand new. But hypothetically, yes, I think you should be able to do that. It'd just be a matter of compiling enough data to support it.
Mm-hmm. Okay. So I guess you touched a little bit about this when you're talking about your friends at UMadison, but like what are some like of the most like promising future research directions you see for like the field of epigenetics?
Ooh. Oh, there's a lot. Um, Well, one of them would be screening for diseases and illnesses related to the epigenome, which was essentially the idea that you just brought up to me and related to grief. And the notion there would be exactly kind of what you brought up. You could take somebody's blood sample or another tissue sample if it needs to be tissue specific. You could then sequence the epigenome. And if you had the data set, you could say, hey, look, the epigenome you have suggests you have X, Y, or Z. And then
Mm-hmm.
you probably have to follow that up, that screening test, with a clinical test to confirm the condition, you know?
Yeah.
And so broadly, what you could be looking at is with just like a finger prick, like just a little lancet, you could actually get a pretty diverse set of um, health implications related to their epigenome. But each one of these health conditions, let's say diabetes, obesity, each one will require a robust data set and quite a lot of investigation in order for you to have the database big enough where you could confidently assign people based off their epigenome. But that, and first and foremost, would be one of the biggest ones. Um, Other things that could potentially be looked at would be modifying the epigenome. So there's some potential right now. They do this for certain cancer therapies where we can remove stem cells, we can culture them in a lab, and then add them back to the person for treatment of whatever illness that particular is. And in that process, too, you might be able to modify the epigenome of those cells that you extracted to further enhance the treatment modality of the stem cells that are captured. Either stem cells, excuse me, or like white blood cells like T cells. You can also do that for cancer. And that's really what I meant. Stem cells are more like tissue regeneration. But in either cases, with those, you know, cell extraction and then lab culture paradigms, you could also apply epigenetics to result in better, healthier cells. So, in other words, what I'm suggesting here, Sid, is that if we can't change the epigenome, if the epigenome is not plastic to behavioral changes like changing your diet, let's say, or other things, maybe we can change it in a laboratory setting, chemically speaking, and then reintroduce those cells back to the person and have them be at maximal functionality.
Mm-hmm. I see.
Um,
Sorry, go
Trying to
ahead.
think what other aspects of it, but blood testing for a variety of different diseases, and that could be really, really big and powerful, is a big one. And then, like I said, modifying the epigenome would be another one because the plasticity might not be there. And then I think too, from a less of an engineering or applied epigenomic standpoint, I think the epigenome can tell us a lot from a public health standpoint, and that's where I do my work. Is
Mm-hmm.
how can we use the epigenome to inform people's behaviors and try to get them to choose better behaviors um, instead? And so, the same thing with like the kind of work that you do related to grief is that another way to kind of use that final point I want to make about the epigenome is to say that one reason why you might want to go to the counseling or deal with your grief is so that it doesn't become a long-term factor that might result in epigenetic changes that could be disfavorable for your health. So, in
Mm-hmm.
other words, not dealing with that problem now might actually result in health problems later down the road for that person. And those health problems might even be inheritable by their children as well. And so that for me would say that's motivation to have better counseling and support for people in grief scenarios. So that way that grief is always going to be a thing, but you don't want the grief to become an adverse childhood experience, if you follow me.
Yeah.
That's kind of what I'm getting at. And
Mm-hmm.
beyond that, we need to be focused beyond just children, although like what you said, the earlier somebody has an adverse effect, the greater the epigenetic change we're going to see. So starting
Mm-hmm.
earlier is better, but really this could be at any point across the lifespan. Addressing these and preventing the changes is going to be critically important. Um, so from an engineering standpoint, we can look at tests using the epigenome. We can modify the epigenome to make more effective cell treatments. And then number three, we can use information from epigenetics to enhance public health and try to motivate people, particularly policymakers, to make changes or to support some of these programs that they think might only be beneficial in the short term,
Mm-hmm.
let's say.
Okay. Well, that's actually all of my questions. Thank you so much for joining us today, and your insight is really helpful.
Of course, Sid. Yeah. Sorry, I kind of got distracted in there.
No, that's
Just
great.
talking.
It's great information. Thank you so much.
So yeah, good questions. Sorry, I don't know more about grief. I'm more of a food person when it was what it boils down to. But I think you're onto something. I think you
Hmm.
got a good idea, and I mean, there's a lot of put it this way, epigenetics is relatively new. Really, kind of the Dutch Hunger Winter study, which is 2008, started that. Now I know you're probably what born in 2008.
Yeah, I was born in 2008.
So maybe it sounds a lot older to you than it is, but that's pretty recent in the context of science. And so
Mhm.
we're kind of just getting to the point where we're not only understanding epigenetics more, but we're going to be able to start applying it in more uh, practical ways to actually address things like disease rates and a variety of other things. But like I said, you got three components of it where it could be utilized. It's a burgeoning field. There's lots of new grants related to epigenetics. And I think kind of the sky is the limit as far as what you might be able to do with it. Um,
Mhm.
yeah.
Well, thank you so much.
All right, so well, I'll let you go then. And you have if you need anything else, you can always send me an email. I'm here for you. I appreciate the work you're doing out there. We always need more public health warriors. Trust me, it's a losing battle usually because people don't want to do the stuff that they really should need to do. But hey, that's life.
Mhm.
And so I don't mind supporting you in these capacities because you're out there doing good, and that's the important thing. And so. Um, Good luck. Let me know if you need an email.
Mhm.
If we keep this up, let me know if you want a letter of recommendation too, and that'd
Of
be just fine.
course. Mhm. Thank
But
you
with
so
that,
much.
yeah, with that, I'll let you go because hey, I'm really busy too, and life is crazy, and I'm sure you got other stuff you want to do too. And so thanks, Sid. I'll just let me know if you need anything.
Okay, thank you so much.
Yep, of course.
Bye.